I was very skeptical maybe 5 years ago. I'm like a reader coming from a liberal arts background. I feel like Sherlock Holmes when I'm reading papers. I was like, an AI is not gonna be able to do what I do. It can do a lot now. What it's gonna be able to do in 5 years, I'm excited. I would love to be probably not replaced, but so I think AI, whether AI is looking at all your data, like your whole genomic data, your wearable data. I currently have AI give me like coaching tips every morning.
What if your DNA isn't a fixed code, but more like a musical score filled with ancient notes and silent harmonies and untapped potential? In today's episode, I'm joined with Dr. Ryan Rossner, Director of Longevity at MiniCircle, and a scientist whose journey from the football field to the molecular frontier has shaped a new vision of what gene therapy can become. On this episode, together we explore how non-viral gene therapy is being used to restore muscle, fight inflammation, and potentially extend health span. What makes therapies like Follistatin and Clotho so compelling for athletes and aging populations alike, the emerging frontier of light-based biology, what it means to tune the body with photons, and how gene therapy, when done ethically and personally, may be less about enhancement and more about realignment. But this isn't just biotech for biotech's sake. It's about co-creating with your biology, listening to the instrument of your body, and stepping into a future that's both scientific and provocative. Let's dive in. Dr. Ryan Rossner, a scientist specializing in longevity research, gene therapy, and the molecular mechanisms of aging. And he's currently the director of longevity research at MiniCircle, a company that is pioneering non-viral gene therapy solutions. So welcome to the DNA of Things, Dr. Ryan.
Hey, thank you, Dr. Jeremy. Very excited to be here.
Yeah. Well, you've got a very interesting background. You know, I was intrigued to say the least, you know, once I came to understand your origin story and dug into your bio, because it's, you know, I don't want to steal your thunder, but it's a mix of intense science and it looks like defense and human performance on the athlete side as well. And now here you are, you know, with this kind of longevity mission, which I'll put words in your mouth by saying something like, you know, live long, die strong. I'm sure that's not your tagline, but I would like to understand, you know, how your journey led you, you know, to this really interesting technology that, well, I mean, has a huge potential, certainly on the first product that I know we'll talk about today, but on future products in terms of, you know, modulating our human genome. So before we start, Ryan, just tell me a little bit about you and how we got to where we are.
Yeah, totally. You know, I think to tell it in story form, when I was 4, my mom got lymphoma, and I could never let go of that experience. So I studied philosophy in college to understand, like, why do things like that happen? And philosophy didn't really answer my questions. It taught me great critical thinking skills. At the time, I was also an athlete. I bounced around the NFL for a bit. And during that time, I had a lot of free time to read. I came across some popular science books that were just like, oh my God, like, not only can science answer these existential questions, it can empower us to redesign the human experience and minimize suffering. So I was just lit up by that, moved to Seattle, switched to molecular biology, worked from the ground up to a PhD with a really sharp focus on longevity. That's how I got here.
Yeah. Okay. So that might be 3 to 30 years that we could spend unpacking that. Okay. You know, philosophy has always had a very special place in my, let's say, hard heart. I started my undergrad in the molecular biology program, genetics, but it was all I could do to resist that pull into, you know, the philosophy, liberal arts kind of degree that was offered at Dalhousie University in Halifax, Nova Scotia, where they have specifically the King's Foundation year, which is a pretty well-renowned, not only philosophy, but liberal arts and even theology.
Oh, cool.
Really amazing thinkers there. Anyway, it influenced me so much so that even, you know, when I chose my PhD supervisor, he was much more of a philosopher than he was a biochemist. He did some revolutionary things, and I got to work with him on those, like deconstructing Darwin's tree of life. And you can see how that very quickly gets philosophical, right? And he did actually, once he finished in science, he got a Herzberg Medal for that work, which is like the Canadian Nobel. But then he went on to become let's say a bona fide philosopher. and we had just some incredible conversations, but it does kind of leave you longing, let's say. And actually, I'll steal a quote from Elon Musk, which is something like, paraphrasing here, don't read Nietzsche when you're a teenager.
Okay.
Right? Well, it's really, I find it very difficult, no matter which philosopher's rabbit hole I go down, to not find myself suffering and in despair at some point. So
Okay.
anyway, it's interesting your own, kind of transition out of that into science. And from what I can, as far as I can tell, in terms of what I've learned from philosophy, it does, as a scientist, certainly make you think different than most scientists.
Yeah.
And I mean, again, of course, that also depends, which scientist you're working with. I mean, I found that, again, evolutionary biology was more kind of my thing, and versus more of a mechanistic biochemist. It was like, I wanted to use that when I wanted to perform, but I needed to know what I wanted to perform at,
Yeah.
So I really love the balance of, there's a hypothesis, there's science that you can do for the sake of hypothesis outcome. And then there's the next level of science that you might do for testing your hypothesis.
Yeah.
But the philosophy is getting to that beautiful question. So, and I'll just share with you also, a similar motive for starting on my end as well is, my mom was diagnosed with multiple sclerosis, and this is something that was influenced by genetics, but also environment, also behavior. And it was like, what is this symphony of life that was going on? And anyway, suffice it to say, here we are on the other side of the simplicity, on the other side of complexity. And we're now talking about, a certain technology that you guys have developed. And, in a very interesting way, like when I check out your website, it seems you guys are very stealthy. And, but yet you've got this revolutionary technology that's underpinning everything. And here you've got a product in market. So maybe we can talk a little bit about like, well, what is the underlying, non-viral dissemination of DNA through this mini circle, and let's see where we go with that.
Yeah, totally. So our gene therapy at Mini Circle is a plasmid gene therapy. So there's no virus involved. There's no microorganisms involved at all in the therapy apart from the production. So a plasmid is just like a small circle of DNA. I did not mean to make the heart emotions pop up. Yeah. So and ours in particular is what might be an umbrella term might be like a minimal plasmid. So it's a very small circle of DNA. For the molecular biologists out there, there's a promoter, a gene of interest, and a terminator. And then there's some other stuff that's necessary. And kind of the magic is in, keeping it small enough so that it works in the human body. We inject it into belly fat with a positively charged polymer that helps it get in through cell membranes, and then it makes its way to the nucleus. It's episomal, sits next to your chromosomes, and we basically set up a little export factory in a few belly fat cells of a secreted protein, follistatin. So you get systemic benefits from just local transfection of a few belly fat cells. We give you more of a gene you already have, follistatin, which basically, makes you stronger, leaner, and probably rejuvenates you a bit at the cellular level.
So basically what you're saying is you can take this, delivery system that we've, borrowed as a byproduct of, well, let's say billions of years of evolution, right? And we've, inserted a specific gene construct of interest here. And in this instance, I guess what we're really talking about here is, lean muscle mass. And so, so for the athlete, I guess you can see how that's obvious why that might be attractive. But then there's really also anybody, again, it's activity dependent, certainly, but really once you get to the age of 35, you're on this kind of, steady downward spiral in terms of muscle wasting, right?
Yeah. Absolutely.
And so you've got this protein that specifically upregulates the synthesis of lean mass. So how is that different from growth hormone versus testosterone, which is like what you see in a lot of these longevity clinics?
Yeah, great question. I mean, I think testosterone and growth hormone are like great examples of double-edged swords. So they definitely improve vitality, strength, energy, ability to heal, but they can also accelerate aging, especially if taken in excess. So follistatin, it's not clear that there would really be as much of a downside to it. Like very high levels of follistatin, I don't think it would be likely to carry the same risks as very high growth hormone or testosterone. And that's one of the advantages of gene therapy, is maybe developing more elegant, better risk reward ratio therapies.
Yeah. When you talk about delivering like this mini circle with the follistatin gene into the belly fat cells, typically where do we find the follistatin synthesized and secreted from?
That's a great question. I'm going to give my best scientist answer and say the short answer is, honestly, I don't know. I don't remember which cells and which tissues secrete follistatin. I do not remember. Yeah, it's a good question.
Well, I'll dive into that. And you know what? We don't have to. We're new age scientists now. And what we've learned is yes, facts are important. But as Einstein would say, is I don't remember my own phone number because that's wasted memory. So we can do real time chat GPT.
Yeah.
And we can get an answer in real time, assuming it doesn't hallucinate on me. We'll have to fact check it afterwards. But
yes.
But yeah, just where that's rendering now in terms of inserting the mini circle into the fat cells. What's the half-life on that? Like how much of this do you need, and what kind of change? What's the delta in the folks that you've been working with?
Yeah, great questions. I mean, we're injecting, we're delivering a very small amount of DNA, so on the order of micrograms. Not all of that. I mean, the literature characterizes the nuclear entry pretty well. A relatively small percentage actually gets into the nucleus and avoids like lysosomal degradation, but it appears to be sufficient to roughly double serum follistatin protein. So that's kind of the order of magnitude of things there.
Mm-hmm. Got it. Okay, so ChatGPT tells us follistatin glycoprotein primarily produced by the liver, but muscle tissue, skin, ovaries, and the pituitary gland. Yeah. So, I mean, it's interesting because this is one of those, cascade, the proteins that has kind of like a cascade of an effect. And so it's really a force multiplier in terms of, anabolic processes. Let's say.
It's often described as a myostatin inhibitor, which a lot of people have even seen like livestock or dog pictures, but it actually inhibits two proteins in your blood, myostatin and activin. And it actually has both let's say pro strength effects and pretty strong anti-inflammatory effects. So pretty cool.
Yeah. And so how long have you guys been delivering this to folks?
I'd say, Mac and Walter started this company maybe 7 years ago, and there was probably like small scale internal testing years ago. And we did a decentralized trial in I'd say about 2 years ago. Mm-hmm. So I'd say about 2 years ago is when it started to ramp up.
Yeah. So you learned about this and you've been working with the company for how long now?
I think maybe about 1 year and 9 months at this point. Okay. And I was involved with the company informally for maybe a year before that.
Amazing. So how has this kind of like made you think about, your life if you had this technology like sooner when you were an athlete or your time in the military? Like what's been going through your head there in terms of the applications?
Yeah, I mean the same thing that goes through my head so often regarding like, let's say, younger people today. We know so much more now than we did. And there's so many therapies emerging. Mm-hmm. I mean, the potential is kind of limitless. So yeah, I mean, it would have been awesome to have, safe access to stuff like this earlier in my life, but I'm excited that we're developing it now.
Mm-hmm. Yeah. And I think something that you've been like developing in parallel to this, or maybe in partnership with other, I think, functional med docs, is this, this notion of medical tourism. And mm-hmm. I guess that's kind of, I think, the the real question that I'm asking is, how are people getting access to your product today?
Yeah, so we have a B2B model. We sell to these medical tourism clinics, and they then can sell it to their patients. So, like stem cells, it's not available in the US currently, but that's our business model.
I see. Okay, so what countries is Minicircle available in?
Uh, Historically, we've been available in Honduras, specifically in Roatan, an island off the coast of Honduras.
Uh-huh.
And I might have to check my sheet here. I'm less on the business end. I think, Dubai and a few other places are either happening now or really developing. So there's maybe about 5 places total that we're either in or developing in.
Got it. Right. And so the Minicircle technology that you guys have, it's not like a one and done with Follistatin, for example. Well, I guess first of all, what would a treatment plan look like for Follistatin? Like how often do individuals do this for typically?
Yeah, that's a great question. I think, to my mind, it's probably like a once a year thing. Mm-hmm. And this plasmid platform is definitely amenable to other genes. Wow. Clotho is one that we're very excited to develop, which could give a cognition boost. So to my mind, it's something that you'd likely do once a year, maybe get like a combination of gene therapies. That's about what I would say.
That's amazing. So basically you guys probably have a menu of, gene therapy solutions that you're thinking about.
Absolutely. Yeah.
I mean, beyond Clotho, can you give a sense of like generally where you guys are going, or just, again, more generally, like what is the realm of possibility here in terms of, well, it seems like it'd be like a DNA tune-up that you would do every once in a while.
Yeah.
Yeah. What's your roadmap look like? I guess the
roadmap. I mean, the roadmap is probably ideally like exponential growth. So that's the beauty of gene therapy. If you master the ability to tinker with the code itself, there's really, there's not many limits if you get really good at that. So, aging or longevity is definitely a big target. Cognition, physical performance, also treating, genetic diseases is really, really interesting to us. So that's more or less how we think about it.
Yeah, I mean, because you could see like, muscular dystrophy as being, an interesting.
Exactly. Yeah. Follistatin has actually had some success treating muscular dystrophy, and we're looking into that pretty actively.
Mm-hmm. I mean, it for folks who are listening who are not molecular biologists or biochemists, this probably sounds like more sci-fi than it is because, well, I mean, I guess what I'm jumping off from is like recombinant insulin, for example.
Yep.
Right.
Totally.
Yeah. And so when recombinant insulin, it's, well, I think it's not a gene therapy, is it? It's actually a protein that you're delivering. Is that right?
Exactly. Yeah. So growth hormone, insulin are both protein hormones that are produced in a recombinant fashion. If you delivered Follistatin in that way, I think the half-life in the body is like 90 minutes. So this is a case where gene therapy is much better. But honestly, for stuff like insulin or growth hormone or IGF-1, gene therapy could be eventually a better option there too, potentially.
Well, yeah, because what I think where I was going with that is you're basically doing the expression of the protein in vitro, in the case of growth hormone and insulin, et cetera. Whereas in this instance, it sounds like more futuristic and more sci-fi, but it's actually more natural because you're asking the body for it to borrow its molecular machinery that it's using already to express proteins in the DNA. You're just kind of providing a template that to genes that might be turned off for whatever reason.
Absolutely. Absolutely right. I mean, it is sci-fi in important ways, and the devil's in the details in terms of making it work and making it safe. But like you said, I mean, we already have these genes, and the foundational research has been done over decades. So sci-fi is becoming a reality in this sense.
Yeah. When does Clotho come onto the market? Can you say, or is that unclear at this point?
It can't come onto the market soon enough is what I would say. But We are working as hard as possible to make that happen. But, it's one of the things that our decentralized kind of fast moving model, we have to hold ourselves to a high standard of scientific integrity. So got to develop it the right way. And so we're just making sure to do that before we put it out there.
Let's pull on that thread a little bit, like independent from, mini circle. Give me a little more of your philosophy brain here. Just let's riff a little and see where we go. Like this notion of decentralized, research, not limited to mini circle. I think in general, take for example, like 23andMe, right?
Yep.
The DNA company, right?
Oh yeah.
Yeah. So they, you could look at them and say, well, that's an instance of decentralization away from, let's say, academia, but it certainly wasn't like a full decentralization because, 10 million people still fall under one umbrella.
Yeah.
But there's these new companies, smaller companies are emerging where leveraging like the blockchain technology now, like getting all of your personal health data.
Yep.
And it doesn't stop there. It could be all of your behavioral data on the internet, et cetera. And
absolutely. Yeah.
Right. So you can imagine it's like, okay, well, I have my data and now I have a mirror and I can see how crazy I am. And now like I get those ads, right? So you get a little more self-awareness, but then you might imagine a future where it's like, so everybody is again, decentralized. It's my own personal data to my family. I'm using it first in my family to grow and nurture good values as it relates to understanding my biology and so on and so forth. And so, but then you can imagine simply, somebody suffering from leukemia or somebody suffering from multiple sclerosis and, seeing that there are these pioneers, these innovators out there and not in the conventional sense either, like not necessarily in academia per se, but as I say, an unconventional upbringing, like a lot of entrepreneurs, for example, who will start an initiative. Like I think about Lorenzo's Oils. I don't know if you remember that movie. It was
no,
it was a rare. I can't remember the name of the disease specifically, but it was basically this couple, their child was afflicted with a fat transport disease, and they were able to prolong his life and prolong his quality of life by actually playing around with different ratios of fats and
interesting
decreasing some of the symptoms. Well, yeah, but the thing is that they weren't scientists,
right?
But it's like as soon as you get diagnosed with something, all of a sudden you become more expert in it than experts. If you take an interest and if you have advocates helping you, because you actually have the experiential side of it. One of my things my track coach used to say to me all the time, when I met him, he was a 70-year-old, Trinidadian sprint guru that taught me more and he took me from being ranked 700th in the country to 7th in the country in 3 months. Yeah.
What event did you do in track?
Oh, I did the 60 meter, the 100, and the 200.
Oh, that's all 7th in the country in that. That's awesome.
University and the 60 meter specifically. So
super cool.
Yeah, he taught me how to have the fastest start to 20 meters. I just couldn't hold it for much longer than 60 meters. But I was a walk-on, and before I met him, I was 700th, right? So
gotcha. Yeah.
But he taught me so much. Like I was training alongside my graduate degree as well, and we would just have we would have so many interesting, philosophical, existential conversations. And one of the things that he would say is, you are the expert in your own body, right?
Yeah.
And that was really what he was teaching me. And, in sprinting, there's zero margin for error, right?
Mm-hmm. Yeah.
And he had this one workout where it was like 10 times 60 meters, right? But the first one you would walk. And then the second one you would walk a little faster. And a little more. And gradually, incrementally, before you even knew it, like he would come up and he'd touch me on the shoulder and he'd be like, run the next one in 6.79 seconds, please. Like asking me to run a time I've never run before. But then you would do it easily because all along the way, you would ask me how it felt. We would do a corrective drill in the moment. And then you would get to this, precision performance that was just incredible. So why did I bring that up? Well, because he helped me in parallel to understanding things, let's say, theoretically, in science, even practically in different lab experiments. We actually helped me experience them personally. So where I was going with this, the Lorenzo's oils or the cancer or the multiple sclerosis, et cetera, and then with, decentralization of information, you can become something, very quick. I'm not discounting the importance of regulatory bodies, et cetera.
No, absolutely not. Yeah.
Okay. So I'm setting all this up to say, you can imagine a future where you've got your own data stored on the blockchain, and it can be instantly mobilized to like all of these different initiatives. And so the speed of innovation, again, we have whole genome sequence data, contextual behavior, all your biometrics, and now AI, right?
Yeah.
So, what you're talking about is like, okay, you've got one solution in many circle, but I can imagine a mosaic of solutions based on
absolutely
what you're presenting with at the time.
Yeah.
And it's fascinating to think about. Some people listening might be like, that's crazy. That's like forever away. I think it could it's 3 to 5 years. Yeah.
It's happening now. So I'm super glad you brought this up. This is something I feel really passionate about. If you look at sports, like whether it's Usain Bolt in the 100 meters or maybe even more extreme in terms of optics, like Alex Honnold climbing in Yosemite free solo or 1,000-pound deadlift.
Yeah.
Athletes figure out ways to do stuff that seems impossible. And if you tried to build those performances solely from PubMed papers, you would not. Like, the athletes are extremely motivated and they figure it out in the field using very live feedback with their own data. I think treating longevity, which is closely tied to disease like this, is likely to produce solutions rapidly. And there's safety concerns, and like in my mind, what we want is both, like the safety and vetted information that comes from academia and regulatory bodies, but then like the innovation that highly motivated individuals like athletes produce. I would argue that Brian Johnson with Blueprint is spearheading something like this and making a lot of his data public. I'm super excited about how rapidly we might be able to make progress with this kind of approach.
Yeah. I mean, it's, something you said there in terms of, even how athletes figure things out. I mean, let's use like the 100 meters as an example, right? Because it's seemingly the most homogeneous event. You start, and then you run 100 meters and you stay in between these two lines.
Yep. And
the point is to get there as fast as you can. And you may be like, okay, that's pretty straightforward. And you might think that everybody's training is the same. It's like, well, I've seen it firsthand that it's not. I have this story that I tell as a tale of two sprinters where I've looked at, into the molecules of these athletes, and the way they get down that line is very different. You've got one that thrives at standing underneath a squat bar with like 500 pounds on it, right?
Yeah.
And then you got another one who's like, he wouldn't squat more than 100, but he loves bounding. He loves more contraction. Working. They're both of them at the Olympic level on the relay team, and one's a starter and one's a flyer, right? And how they train is so different. And so having that kind of, I don't even like to call it a recipe, because you would never follow somebody else's recipe. I think that certainly it's like, okay, success leaves clues. What aspect of my body is like theirs? You know what I mean? So like, I'm definitely more of a push sprinter, for example.
Okay.
So I thought, until I started breaking everything. So I would tend more to weights, but I actually learned later, this is working with Garney, he's like, no, actually you have more, potential at the levers than you might otherwise think. And if we learn how to get the right muscles firing, you don't have to lift all this weight. So
interesting.
So there's kind of these thinking of the human as a system, right?
Yep.
And so what's a system? A system is like it's made up. It's a paradigm, right? It's what's a paradigm? Paradigm is a map. It's a way of seeing the landscape, but it's not the landscape itself. So I think when we get into this space with decentralization of data, the application of, let's call it systems biology, and i.e. therefore paradigms, it's exciting to think that as more people collaborate, and again, I'm speaking in the ideal scenario, you get this kind of spiraling up of understanding
Yeah.
about the human condition. And then you learn as an individual human, well, there are different versions of you at different times in your life and different therapies. And this one worked when you were younger and it's not to be used when you're older. And so even that future is really, really, really interesting.
Yeah, definitely.
Yeah. So when you think about, mini circle and it in its most kind of ideal, hopeful future, what is it, given everything that we just talked about, how does it kind of fit into, I guess an auxiliary or a supportive system in sync with like kind of this digital twin type of possibility that's emerging with AI and technology?
Yeah. I mean, you've mentioned genome sequencing at least twice, I think, during this conversation.
Mm-hmm.
I'm like evangelical for people to get their own genomic data. I think that's a great starting spot. Like, know what you're working with to begin with. And then, at MiniCircle, our passion is really to make this accessible, both by bringing the price down and by educating and empowering people. So, I think there's a lot of ways it can fit in, and we want to make that accessible to everyone so that people are empowered to, shape their own identity from the molecular biology level, from the ground up.
Yeah. Well, you can imagine, like, once you've got this standard or set, let's say, of biomarkers, and you consider them in concert, and it's obviously like a temporal analysis that you want to consider. I think we have to define like what is health for that individual at that time.
Sure, yeah.
Because health for me when I was 10 is not the same as when I'm 40. It's different.
Of course, yeah.
And then, what is the supplement isn't the right word. Fertilizer isn't the right word. It's kind of like, I don't know, it's like an elixir in a way. That I know you're focused on longevity, but I mean, I see so many applications in terms of like, and this is probably getting way too far ahead of ourselves and too sci-fi, because I know you guys don't live here right now, but you could even think of it as in terms of, well, at different developmental stages whereby you need support of not these specific products, but future products. And then how much of it should be, exogenous versus endogenous, I guess is kind of the, topic of philosophical questions is, like what is the ideal playbook look like in terms of an individual being able to leverage technologies like these?
Yeah. I mean, I think to me, one ideal is, just maximum individual empowerment. I think people should be, I'm a big freedom guy, so I think people will land on their own different personal ideals, but, the more, the better informed they can be and the more tools they can have at their disposal, I think the better outcomes we can all get.
Yeah. I think about it now. I had one of my guests on previously. His name's Dr. Miles Nichols, actually from.
Oh, yeah.
You know him. Okay. So
we know Miles.
Okay. Yeah. I mean, he's building something, to the degree that I had just described, this whole kind of live long, die strong protocol. But the thing that I like about the way that he's doing it is he's taking the expert knowledge of a practitioner, specifically in this instance, like a functional med doc, and he's providing that expertise in that frame with all these different, cutting edge technologies. And so there's this, kind of liminal space that he's living in where it's like a lot of these things haven't been done before. Well, in fact, none of them are because in every instance, every person's care plan is different, right?
Mm-hmm.
So he's leveraging the information technologies and the AI, but what he's bringing to bear is the expertise and therefore the critical contribution, which is knowing what question to ask, right?
Yeah.
So it's like, okay, I have my intake form, and then based on all the data that you've given me, I've optimized my decision tree call it differential diagnostics isn't really the right term, but if I say it, I think you know what I mean.
Totally.
Because you're really trying to go even, it's like almost a layer down below differential diagnostics because you want to get to the root cause.
Yeah. Yeah, definitely.
And provide a systems level of analysis. So
absolutely.
So yeah, I mean, I think insofar as the expert practitioner, and if you think about the different disciplines that you've encountered, what's the spread? Like, are you seeing, you see longevity docs? Well, what are they? Are they MD? Are they specialists in some fields? Are they functional med docs? Are they chiropractors? Like, what are you seeing in terms of the practitioners that are interested in what you're offering?
Yeah, I think we see interest. We see interest from a lot of different people, including a lot of different healthcare, providers, different types of doctors. I think, I'm not sure the term you used, but certainly MDs, but whether it's functional medicine or integrative medicine, I think we see a lot of interest from that group. Mm-hmm. Yeah, for sure.
Amazing. So when you think about, the ethical considerations, I mean, you're not operating in the US, so you guys, I'm sure, anticipating some kind of pushback, or I think when you're in this space, well, even genome sequencing had and still has a ton of ethical considerations. So I'm not.
Yeah.
What I'm interested to know is, what do you see those considerations, like what might they be? What ethical concerns do you foresee with kind of widespread access to gene therapy? And feel free to be as sci-fi as you want, because for context, I don't know if you remember that movie like Gattaca.
Yeah, of course. Yeah.
Right. So what does it mean? It's like, okay, maybe I'm not changing my code yet. That's next, by the way, with CRISPR-Cas9. But
yeah,
you've got this step in between where people, once they get on the side of getting over disease and optimizing longevity, you can see the temptation to become so-called superhuman, whatever that means. So what are you thinking in terms of the ethical considerations?
Yeah, I think the first ethical consideration is safety.
Sure.
And then beyond that, in terms of enhancement, anything that somebody's doing to the like midlife or in vivo enhancement, it's a risk to consider that you could create some polarization of society with, like, if only some people have access to extremely effective therapies that can make you superhuman. Again, we're working towards widespread accessibility for these therapies at MiniCircle. But then that's, gene therapy for adults. Once you get into germline engineering and changing things at the, embryonic stage, I think that's a whole other can of worms. But the thing is, progress is good, and it's really best to just deal with these ethical challenges, because the potential is much greater, I think, than the risks.
Yeah. So you said you had experience previously in military or defense, is that right?
Yeah. So I was a civilian. I did a postdoc where I was a civilian contractor for the Air Force working on a DARPA project.
Gotcha. You're right.
Human hibernation was the project.
Human hibernation. And what did you guys find?
I would say it's very doable. There's like a pretty well-defined group of hibernation triggers, which are kind of some usual suspect molecules. And then there's some downstream effectors that are very cytoprotective molecules. To my mind, if you apply a cocktail of the triggers and maybe boost the downstream effectors, I think it's very likely you could get therapeutic or hibernation for other purposes in a human.
Wow. So you could opt to be like a bear and just kind of check out for 3 months in winter, or?
So that might be like the deep space travel scenario where you're rotating a crew, but you might think about it in terms of a metabolic spectrum.
Yeah.
So what if you could perform on a daily basis while running at a lower metabolic rate, or maybe have a more efficient metabolism, like lower input, cleaner burning, higher yield? It's really about tinkering with metabolism. So it's pretty fun.
It's really interesting. I remember I was captivated by there's some species of, I don't know if it's a sea turtle or I think it's a freshwater turtle. They just basically live up, right? And they go pure anaerobic. right?
Yeah. Th-
And
yeah. There are a ton of animals and other, species from other kingdoms of life that can do it. Like humans are really limited in our ability to lower our metabolic rate. But there's a ton of examples and a lot of different ways it's accomplished in nature. Yeah.
there's a version of this. I don't think it's the same thing at all, but maybe you can tell me because you're expert. But like I feel like there could be some parallels here where, in particular, just playing with diet and nutrition, you can switch over from, say, a responsible carbohydrate diet. So what do I mean by that? Kind of like your ratios of 40, 30, 30, glycemic
Yep.
index balance, et cetera. Anyway, the point is your body's anticipating. That's what's so cool is you've got like residual enzymes in your body. So in a way, it looks like it's anticipating what's coming, right?
Mhm.
Because
Yeah.
life of those enzymes are, long enough to be relevant to the next meal, let's say. But then you get into a point, and I found this fascinating, is, switching into something that is, more ketogenic, for an example. And
Yeah.
you hit that period and there's like a big emotional tug, right? As the kind of the final phase of your blood sugar, at least exogenous blood sugar is kind of regulating more to gluconeogenesis versus again, exogenous sugar source. But
Absolutely.
then once you get to the other side of that,
Yeah.
it's like well, I mean, the ketone bodies that your brain is using, the side effect too is if you're cutting out carbs in general, what you're doing is you're cutting out by default, all the refined carbs. And so you're eliminating all the issues to do with, any of these like kind of contaminant, preservative types of things. So you're losing a ton of inflammation.
Yep.
And then switching into like carnivore specifically is like, well, protein's not very efficient, is it, as an energy source, right? Yeah. So,
Yeah.
and then I'd go into like full fasting, and then you get to the autophagy as like kind of a final phase. And I'd come to the end of some of these kind of, self-designed things feeling like I never need to eat again. Right.
Yeah, that's the dream, right?
Yeah.
I mean, just like a perfect self-sustaining being. Maybe you're living on light or something. Yeah, I mean, dream big. Like I constantly fantasize about this and work in my spare time on the weekends to like, hack my metabolism. I go for this weekend. I did a long run, no food, not too much water either. Yeah, I mean it's a wonderful dream. Like, yeah.
Well, it is interesting when you just think about how impractical it is for just say for entrepreneurs, or just like, you kind of just, not that I'm condoning this, but you can see days where you're working, not too many in a row, by the way, but like maybe you're working 20 hours straight, and you can pulse that in a way that it's sustainable, like using first principles in human performance, like athletes and periodization, et cetera. So there's a lot of really interesting things you can do through your behavior, artificially change your environment, but then to support the right profile of, supporting nutrients. And so this is where I was going by asking you about your time in defense, was exactly on the space travel side of things, right?
Yep.
And then you already went there on like, I'm not sure if you said it or didn't, but it took my brain there on the idea of like even like photosynthetic capability.
Yeah. No, yeah, I said it. Yeah. I mean, it just, I not to get too woo-woo, but like light, I think about light a lot. Yeah. I mean, I actually worked with a guy at the University of Washington who was developing light activated mitochondria to put in. The goal was to make it a human therapy. So you would literally generate energy from light instead of the old fashioned way. So yeah, I mean, we should dream big scientifically. These problems aren't necessarily impossible to solve. It's just nature didn't have an incentive to solve them in the human body, apparently, or long story short, did not solve them, but it doesn't mean they're not solvable. Yeah.
It's funny you say the old fashioned way, but maybe light is the old fashioned way, because you look at the mitochondria, right? And well, they already do have cytochromes, and they're using that kind of deep red light therapy, I think, in like some chiropractic clinics, really to like activate the point of injury to turn on the energy production there.
Yeah.
And like really down by activating the cytochromes is like better potentiation of the intermembrane gradient there, which is where the ATP is made. So
I mean, it's this is when I can get free, when I can get my work done for mini circle stuff and have some free time where I still have energy. Looking at the biology of light is one of my top priority side projects. Like I would say it's underdeveloped and it's just absolutely fascinating.
Amazing. It's funny that I spent time in Woods Hole, Massachusetts at the marine biological labs there. And the enrichment experiments that we were doing, that I was doing, was looking at different spectrum of light. Because the whole course was about developing protocols to cultivate the unculturable majority, which is, if you look at a soil sample, it's like 99.9% of the bugs that live there, you can't grow them because they're so synergistic, right?
Interesting. Okay.
So the whole course was about, okay, well, how do you isolate these things? And anyway, I took the approach like I wanted to use light. So I created different media that were nutrient poor, minimal media, so that light would be the priority thing that they were. It was like those that could best use light and the minimal media would be the ones that would survive. And I
totally
went across a bunch of different wavelengths, but it was a bad experimental design because I only had 2 days to do it. If I had 2 years, maybe it would have been better. But
totally.
But there was another guy. His name was Remy at the time. So he was at Harvard at the time in their interdisciplinary program. He was a wild thinker. I really liked him, and he was doing a similar experiment. But what he was doing is he was like perturbing the light like every hour. Anyway, both of our experiments came from a good place, but they were poorly designed. So what we ended up doing, this will bring it full circle, is we ended up using metagenomic techniques to basically extract whole genome sequence DNA and harvest plasmids. So we actually we digested all the linear DNA, let's say, and we enriched the plasmids from the environment, and then we inserted the transposable elements to then put selection markers on them and then grow them. And we actually isolated a number of mobile genes, which by the way is what my graduate work was really about. It was more the molecular ecology of a mobile gene pool as assessed using these metagenomic techniques. And
super cool.
And it is, because what you see is there's these like super highways of genes being exchanged, not only between non-pathogenic and pathogenic bacteria, but even between bacteria and human, if you consider, the endosymbiotic event of mitochondria to the eukaryotes. And so those genes are jumping back and forth. And okay, but I did want to circle back on your one light thing, because what fascinated me was this sea slug. You've probably seen it. Okay. It grows basically an appendage that for all intents and purposes looks like a leaf. And what it does is it eats cyanobacteria.
Okay.
Or no, it eats algae and it harvests the plastids, and it subsists on light. So it's a snail and it lives on light.
I did not know that. That is super cool. Yeah.
I had to give you that, given all the time that you're spending on light. But to bring it back to this kind of space travel thing, it's like, well, how well can we harvest light? And we know we can, because we make plants and plants feed them. And it's like, well, can we get rid of the middleman for?
Yeah, exactly. Yeah, exactly. Yeah. I would be okay with green skin if it meant I could live on light.
Exactly. Right.
Yeah.
Okay. So this is, I mean, I'm going to make the leap since we're already there. It's like, hey, maybe that's why all the aliens that we've been taught about in science fiction are green, is because they're not really space travelers or time travelers. Maybe they're humans coming back in time. Right?
I think it's important not to censor hypotheses at the hypothesis generation stage. So same thought entered my head. Aliens are green. Maybe this is why. Yeah, it's a fun idea.
Well, I mean, just the whole idea of astrobiology. And by the way, I'm pretty sure that was pioneered around the time of Carl Sagan.
Okay.
Who, he did wrote the Cosmos, and
right.
Neil deGrasse Tyson. He actually mentored Neil deGrasse Tyson. And I think he wrote the book also, Contact, with Jodie Foster. I think that's true. Don't hold me to it. But it really is that kind of far out thinking. Well, even Carrie Mullis is another one. It was like, went for a surf one day, and well,
yeah.
With a certain cocktail of
I think Carrie Mullis. I think there's like an electric raccoon. Was involved. He had a vision of like an electric raccoon that helped him understand or develop PCR. I mean, science is. the actual history of science is much weirder than, I wish science would talk about this more because it makes science that much more fun, like, yeah.
Well, so coming back to one of the things we talked about, which was, right, the cure for diabetes, right? Like, so Banton and Best, they isolated, I think it was the insulin they isolated originally was from pig.
Okay.
And they did it, but they didn't write it down. They messed it up. And they had to do it again and again. So yeah, the science is present, even the way the scientific papers are written.
Oh yeah.
Are not true, right? I mean,
no.
And every scientist knows that they're written to be consumed and hopefully facilitate knowledge transfer.
Yeah.
But you're like, I still think it's like the journal of negative results, the journal of failed experiments. You gotta have that in,
I agree very strongly. I mean, I'm not even sure that the incentive structure is optimized for scientific communication in terms of publishing. Like, I think it's optimized for publishing and career building to an extent that is considerable. Like, if I had a dime for every scientific paper I've read where it seemed like it could have been better written if the goal was to communicate their findings to me, a fellow scientist. So not only that, but I strongly agree. I mean, negative findings, also just anecdotes, like maybe a non-peer-reviewed, periodical with wild anecdotes from behind the scenes. I mean, non-scientists might enjoy that. Scientists would almost certainly enjoy it, and we might learn stuff from it for sure.
Well, it exposes our humanity, I think.
Yeah. Yeah, it does.
Like for me, mathematics became really interesting once I understood the philosopher behind it, right?
Yeah.
Once I understood the personal struggles of their lives and the challenges of their time, and and that there was a turning point for me in academia, probably around my third year of undergrad, where I was like, oh, like there was a time when, like, early in undergrad, it was like, oh, I don't want to read those case studies on the side; those are boring. I don't care about the methods. Da da da. And then there became a time where I actually started doing research. Actually, well, gosh, more plasmid relevance here. I studied the yeast 2-micron plasmid that is in yeast Saccharomyces cerevisiae, which is like basically the lab version of E. coli for.
I come from a yeast lab, yeast and worm lab. Yeah.
So we looked at what was it? It was a 2-micron plasmid. It's got this like tiny plasmid that doesn't have any genes on it that would suggest any evolutionary selective advantage. And so it's like, well, why is it there? And anyway, my job was to like figure out why it's there. And anyway, we did a whole bunch of really cool cell cycle synchronization type of studies to reveal that, well, actually one of the proteins on it is phosphorylated at a certain time in the cell cycle, and it interfaces with these other transcriptional regulators downstream. And so actually, as it turned out, there was some kind of role it had to play in cell division, although it wasn't super obvious. But that's around the time I met my supervisor at Ford, where I was like talking about, hey, isn't it amazing that there's this gene or there's this plasmid and it doesn't even have any function, and there it is. And so this is again Ford, who would be a big fan of Richard Dawkins and the selfish gene, and I was there, I had a flu at the time, and he's like, you think that virus that's in you right now, like, that's good for you? Like, how's that function to help you as a human? Kind of. So he's like a very Morpheus Neo moment. He's like, you think that breathing, right? So So there again, this comes back to like paradigms and mental models that we impose on biology. They're better than they were 1,000 years ago, maybe. But they're not true. 100%. Yeah.
My mentor used to tell us over and over again, your model is wrong. It doesn't mean it's not valuable. You should keep presenting models, but it's wrong. Yeah. I mean, to touch on something you said a couple minutes ago, humanizing scientists, I think it doesn't detract from our credibility. I think it's actually helps bridge the gap between scientists and non-scientists. I really think that could go a long way.
That's amazing. Why don't we use that to kind of segue into the last question there, which is, if we did that, what would Well, what did I say? I think the question I was something like, yeah, what does longevity and human evolution look like in the next 5 or 10 years? And totally, like, I will not hold you to your answer because who the hell knows? But anyway, since we're already in that kind of riff,
Yeah.
what do you see in the next 5, 10 years? Keeping in mind that everything that Elon Musk is doing, if he's successful.
Yeah. I mean, I was very skeptical maybe 5 years ago. Like I'm like a reader coming from a liberal arts background. I feel like Sherlock Holmes when I'm reading papers. I was like, an AI is not gonna be able to do what I do. It can do a lot now. What it's gonna be able to do in 5 years, I'm excited. I would love to be probably not replaced, but so I think AI, whether AI is looking at all your data, like your whole genomic data, your wearable data. I currently have AI give me like coaching tips every morning So I think AI is a game changer and will become even more of a game changer. I think the pairing of affordable whole genome sequencing, which is like massively underappreciated, and the emergence of accessible gene therapy is also going to be really big. And then, kind of other, more powerful therapies, whether it's, epigenetic rejuvenation or, other things of this nature. I think that's kind of, and maybe like brain computer interface stuff, because I really think our cognitive bandwidth is kind of a limiting factor for us. Like you can only get so smart. So those are the things that I see really having the biggest impacts in the next 5 years.
Amazing. Sorry, the preamble that I sent you ahead of time is what I like to typically route out the podcast with. And so there are three questions that, I like to ask. Number one is, if this was your last podcast, and what would you say? Like, I don't know if you're a Johnny Cash fan, but that's the question that was asked.
Johnny Cash, yeah.
So it's like, well, what was the last?
Yeah.
Well, if you had one song to play, what would it be? Is Folsom Prison. But so what's your Folsom Prison? That's number one. Number two is like, who's your audience? Like who needs to hear this? And then number three is, I guess, who would you nominate to be on the show?
Yeah, I think so. The first one, I would say, I would suggest to people work in harmony with the natural world and our natural biology in order to transcend it. Like we do not appreciate how the degree to which we are limited and the degree to which our physical biology is not aligned with the heights our spirit is capable of. So I would say that's what I would say to people. The audience, I would say, is anybody who feels limited by their biology, whether it's disease or whether they want to do something, great that they can't do, like whatever it is. That I would say would be the audience. And then as far as somebody to have on the podcast, I had an answer written down, but based on this conversation, I'm going to change it. So there was a guy, I don't know this. I'm just going to say, so there's a guy at University of Washington, Gerald Pollack. Okay. I am not capable of assessing the credibility of his work, but he is a biology of light guy. And he has a couple really cool books. But he is like, that's what I would suggest. Gerald Pollack, Biology of Light, could be a super interesting conversation. Yeah.
Illuminating. I will see if I can track him down.
Yeah.
Amazing. Okay. So Ryan, just before we wrap, how do people find you? How do people learn about Mini Circle? For people who want to kind of reach out and look to harmonize, look to learn more, how do they best find you?
Totally, yeah. Mini Circle, we have an online presence in all the ways one might expect. So minicircle.io is our main website, and minicircle.clinic is our clinic website. We're on Instagram, X, LinkedIn, as I think typically as Mini Circle DNA. I'm on Instagram as RJR PhD. So please feel free to look us up, reach out to us. We're around. Amazing.
Well, Ryan, I can't thank you enough for joining. I'll say it again. The conversation, was certainly inspired by the source, the light, and it was illuminating. So thank you.
Yeah, thank you, Jeremy. This was awesome.
So what if the genome is a draft, not a destiny? In this conversation, Dr. Ryan Rossner didn't just unpack the technical advances in plasmid-based gene therapy. He reminded us that behind every biological tool is a philosophical choice. Will we chase perfection, or will we learn to partner with our potential? We talked about decentralization in medicine, the ethics of accessibility, and how tools like red light stimulation, fasting, and N-of-1 experiments are helping people tune their biology, not just hack it. And now, the thread is yours to follow. Here are 3 questions to take with you. So how about this? Number 1, what aspects of your health do you treat as fixed when they might actually be flexible? Number 2, how can you start gathering feedback from your body and not just imposing protocols on it? And number 3, what would it look like to view your DNA not as limitation, but as a living invitation? So you can learn more about Ryan and Ryan's work at minicircle.io or by following him at Ryan Rossner. Until next time, stay curious, stay reverent, and remember, DNA isn't a script to obey. It's a language we're all still learning to speak.